will have a longer time of asymptomatic time than homozygous individuals. Homozygous at one locus or homozygous at 2 or 3 loci. And of course, this can be easily explained by the fact that heterozygosity allows presentation of a higher number of HIV peptides, a larger range of HIV peptides to CD8 and CD4 T cells, and therefore allows individuals to better control infection and to prolong the time for viral mutants to arise. You know that one major problem in HIV infection that is the high rate of mutation of this virus, which is a mean for the virus to escape immune response. So apart from this heterozygous advantage, there are some alleles that confer either a protection to the development of the AIDS disease, or on the contrary, which accelerate the progression to AIDS in HIV-infected subjects. And these data have been now largely replicated in many different cohorts, and they all confirm that there are two alleles, B27 and B57, that are strongly involved in a delay in AIDS progression, which means that these two alleles confer protection from the development of AIDS disease in HIV-infected individuals. By contrast, B35 accelerates the onset of AIDS. So how is it possible? There have been now quite a lot of studies well demonstrating that the functional basis for this association, in particular for the protective effect of B57 and B27 alleles. And they all conclude that B57 allows B57 can bind, in fact, a large set of peptides because it has a broad cross-reactivity against bound peptides, so it can bind different peptide variants that occurs, for instance, from viral mutations, so that there will be always at least one HIV peptide that will be presented by B57, and the virus will therefore have many difficulties to escape the T cell response. For B27, it seems to be a quite different mechanism. B27 is known to present a conserved epitope in an important antigen of the virus, which is derived from the GAG protein. And this P24 GAG epitope is extremely important for the virus to survive. If there is a mutation in this epitope, then the virus is not powerful, so it cannot infect anymore. So B27 presents a very conserved B24 GAG epitope, which is under structural constraint to remain intact. That means that the virus has no interest to mutate this epitope, otherwise it is not functional. And I would like to show you some data of a recent study which showed the mechanism of the B57 associated control of HIV infection. This is from the Australian group, where they studied all the using algorithms that predict for a given peptide, a given allele, which kind of peptides can be bound to this allele. You can find these algorithms on the net. And so these algorithms predict whether a particular peptide will bind a given HLA molecule. So using this prediction for B57, they test the whole human proteome, that is the combination of all potential self-peptides in the human, which give rise to approximately 10 to the 7 unique peptide sequence derived from this whole human proteome. And they observe that among all these sequences, 70,000 were predicted to bind to B57, which is protective, which prevents from progression to AIDS, while a much larger number was predicted to bind to non-protective allele. For instance, it's shown here for B701, but it can be even a higher number for B35, which, as I showed you, is an allele that favors progression to AIDS. So it means that the peptides which are encountered by B57 restricted T cells in the thymus. You know that T cells are educated in the thymus. And these T cells in the thymus will encounter a smaller number of peptides presented by B57 than T cells restricted to another allele. This is rather counterintuitive at the first view, but in fact they deduced and they made in vitro experiments to show that these B57 restricted CD8 T cells, although they have been selected against a smaller number of peptides in the thymus,